Congratulations, you've made it all the way to the very last section of our lecture. I hope by now you've spent some time looking outside at the pretty snow, that you've curled up with a cup of hot cocoa, and I promise that we'll get through this last section quickly so that you can actually go outside and enjoy it. We're going to talk about bulk transport across the plasma membrane, so not one molecule at a time, but a full sample at a time. When you have transportation out of the cell, we call this exocytosis, and transportation in, endocytosis. I like to remember this because exocytosis sounds like exit. Small molecules in water enter and leave the cell via those transport proteins, at least most of them by transport proteins. But large molecules like polysaccharides and proteins often cross the membrane in bulk via entire vesicles, but this bulk transport always requires energy. In exocytosis, those transport vesicles migrate to the membrane, fuse with it, and then release their contents outside of the cell. We've seen this before, but now I'm giving you a name for that process that moves all the way from the ER through the Golgi and to the plasma membrane to release non-membrane bound secreted proteins. We call this exocytosis. And there are videos of each of these that you can watch online. In endocytosis, the cell takes in macromolecules by forming vesicles from the plasma membrane. This is the reverse of vesicles we've seen before that form from the Golgi. Endocytosis is the reverse of exocytosis, but it involves different proteins. Remember, we're talking about a different source, different membrane entirely, and frankly, different stimuli, different things that are going to cause exocytosis versus endocytosis. There are three types of endocytosis: phagocytosis, which we call cellular eating; pinocytosis, cellular drinking, which I sort of remember because I do enjoy a nice Pinot Noir on occasion; and receptor-mediated endocytosis. Here's a picture of each one of these. Note that phagocytosis is going to be the uptake of these large molecules, such as this here, into a food vacuole, which we've seen before. As you know, these food vacuoles are going to merge with the vesicles lysosomes, which have an acidic pH caused by proton pump, which you've now learned about, and together they're going to digest this protein or other substance down. Pinocytosis, which is our drinking, is going to be taking up solutes and other components from the surrounding liquid in this extracellular space. This occurs in a slightly different fashion. These are pits, and they're coated by a protein. They have a very nice regular structure. Note that phagocytosis doesn't have as regular of a structure, and this is actually going to lead to a coated vesicle. A very similar kind of vesicle is going to form from receptor-mediated endocytosis, but the stimuli will be different. The receptor-mediated endocytosis is caused when the receptors on that plasma membrane recognize some sort of a signal that triggers this change. Here's a video which is
Watch as one
actually
cell
going
is
to
consumed
show
by another cell in the
phagocytosis.
process known as phagocytosis. In this video, a yeast cell, red, is being consumed by a larger cell of dictyostelium, which is expressing a fusion of green fluorescent protein with coronin, an actin-associated protein involved in phagocytosis. Notice the expression of the coronin protein fusion where the dictyostelium cytoplasm is surrounding the yeast cell. Credit: Gunther Gerisch, Max.
In phagocytosis, a cell quite literally engulfs a particle, and your immune cells are sometimes even capable of engulfing cells, entire cells themselves, that are as large or larger than the immune cells. Other times, they're just smaller food particles. It depends on the cell and the purpose. But what you'll note is that these are not structured, beautiful circles that are occurring in phagocytosis. The goal is to engulf the entire piece of food on the outside. And remember, in phagocytosis, this vacuole, this food vacuole, is going to merge with a lysosome to digest that particle or food source. Here's a picture of an amoeba, which is going to take up an entire green algal cell. In penocytosis, the molecules are dissolved in droplets and taken up when the extracellular fluid is gulped up, drank. And so the fluid is taken in, but we're not talking about full food particles. Compare our previous phagocytosis to these much smaller sections, these little penocytic vesicles forming here. Perhaps you can note, using your skills at looking electron micrographs now, that there's a thicker section of dark. There's a single layer here for the plasma membrane and for here, but this gets a little bumpier and thicker. That is due to the protein coating around that pit. In receptor-mediated endocytosis, binding of a specific solute to the receptor is what triggers this vesicle formation. Note here you can see again this electron micrograph with the continuous plasma membrane moving in all through and around this forming vesicle. On the outside is this little more bumpy but also electron dense section here, which is actually going to be the coat protein of this pit. We call it clathrin. Human cells use this receptor-mediated endocytosis all the time. One of the major components is the recognition of cholesterol. When cholesterol is found outside, for example, in your blood, it's taken up and used by cells as in the formation of new plasma membrane or other membranes. As you know, cholesterol can insert itself into that phospholipid bilayer, and it can actually influence the fluidity and the stability of that fluid nature for a phospholipid membrane. Cholesterol in the blood is found in particles called low-density lipoproteins. That's where LDLs come from. That's what the name means. So, as you might imagine, lipoproteins are composed of both lipids and proteins into structures that can be used. Some individuals with the disease familial hypercholesterolemia have missing or defective LDL receptors. So, they still have the cholesterols. We get the cholesterol from the food that we eat. But the receptor on the surfaces of their cells that's supposed to signal the uptake of that cholesterol out of the blood and into the cell where it can be used are broken. And so this causes an increase in the amount of cholesterol concentration in the blood. Some individuals who have this increase of cholesterol in the blood can develop atherosclerosis. It's a buildup of molecules inside of the blood vessels that restricts blood flow, and it causes heart attacks. So, don't forget that we have a variety of different kinds of endocytosis. Exocytosis exits the cell, but these kinds of endocytosis bring resources from the outside of the cell in. Cells such as bacteria are taken up by other cells, like white blood cells. But can you tell me which of these mechanisms is responsible for this particular kind of endocytosis? The answer is phagocytosis. Remember, phagocytosis is going to be that actual eating of large or other food molecules that can be digested up for nutrients. Penocytosis is drinking. Receptor-mediated endocytosis is still going to be those coated vesicles like penocytosis, but with triggering from specific solutes in the environment. Exocytosis is an exit. It's not going to be involved here. And with that, I will leave you with a review of some of these important concepts. Please don't hesitate to send me an email if you have any questions about the material that we have covered today. You still have a homework that's going to be due before class on Monday that covers all of this material that we've been working on. It's really important that you don't fall behind, but please don't forget that you do have an exam coming up next Thursday. You're going to want to be studying for that exam. And don't be afraid to start incorporating that knowledge towards our new ideas. That'll help you do better on the exam when I'm asking you to do things like connect ideas. You'll have to understand more. And again, I'll emphasize that I will be available by email all day. So send me an email if you have any questions, and we can still find time to get together and talk before the exam if you have any concerns. Have a great snow day, guys, and I will see you on Monday.